Welcome! My name is Astrid van der Brandt, and on behalf of my colleagues from Eindhoven University of Technology and Wageningen University, I am pleased to present to you our design study that led to PanVA, a visual analysis tool for pangenomic variant analysis. Before we talk about the design, let's explain what variant analysis is and how it's done with pangenomes. Understanding an organism's DNA sequence makeup or genotype and how it links to detectable characteristics or phenotype is important in many biological domains, such as human medicine, microbiology, virology, and plant breeding. Traditionally, genomics researchers in these domains try to identify genetic elements contributing to these relations by comparing the sequence of the basic units or nucleotides to a reference genome for that species. Then they assess their impact with annotations and metadata. Nowadays, the growing number of sequencing output has led to the insights that comparisons against a single reference genome can introduce biases. One major bias stems from the fact that the reference is often derived from one or a small set of individuals of a species, and therefore it ignores the natural diversity that may be present within a species. For example, regions that are absent in the reference are missed. Secondly, many organisms are diploid or polyploid, meaning that they have two or more copies of their DNA, but they are represented by a single genome sequence, ignoring the true copies and possible variations among them. For these reasons, many species are now represented by multiple reference genomes. To perform comparative analysis across all these genomes, the field is moving to pangenomes, which are abstract, often graph-based representations of the genetic material of multiple related organisms. Pangenomes can be constructed in many ways. We make use of hierarchical graph-based pangenomes as constructed by PanTools. These pangenomes allow for a holistic analysis of the diversity of sequences, with which is possible to detect more variants and make easy cross-linking to annotations and metadata features. So the motivation for this work is that pangenomes are increasingly important in various fields because they effectively represent diversity. But their data and analysis complexities also hinder human interpretation and insights. Human inspection is still needed to assess the biological relevance. To support genomics experts in these fields, we developed a visual analysis approach that uses tailored visual encodings and interaction strategies to explore and interpret these variant data. To understand how to create an effective visual analysis design, we first delineated for what kind of tasks and analysis goals, that is why, genomics researchers would need it. We ran a formative, user-centered design study with active involvement of genomics experts from industry and academia. Our approach has been threefold. First, we conducted semi-structured interviews and observation sessions with 6 researchers. This helped us to prioritize goals and tasks for pangenomics visualization. The main outcome of this was that small-scale sequence variant analysis is one of the major goals in the current workflows for crop and pathogen research. A second approach of this process was to create a visualization opportunities workshop. We held this CVO workshop with 15 new participants. Here we gained deeper insights on the current analysis workflows for high-level goals and also the tasks and design requirements related to them. We found that genomic scientists want to analyze small-scale variants for either open-ended exploration of genotype-phenotype relations or for more specific marker discovery, where exploration is done with the aim of finding sequence variants that can distinguish data into groups. For both goals, the tasks revolve around locating and browsing interesting regions of conservation or variation to then explore those regions in more detail, considering the sequences but also annotations and evolutionary relations and metadata. And lastly, comparison and summarization aspects to broader consider the genomic context. For more details on the task analysis, we refer to the paper. Lastly, we also held recurring bi-monthly presentations with all collaborators during video calls and face-to-face meetings. These sessions helped us to reflect on the process and ensure that all proposed ideas would meet the diverse needs of genomics researchers. Guided by the task analysis, we translated relevant pangenome information into meaningful views for genomics scientists. The design process has been iterative, where we started with low-fidelity sketches and prototype of mostly familiar encodings to test out what works. The current design consists of 3 components, of which A and B are merely for data selection. Therefore, we will focus on view C, the locus view, as this is the most relevant view to our contribution. The view integrates 3 subviews to explore a region of interest in more detail. The sequences are shown in the middle in the multiple sequence alignment, and the gene model annotation track is aligned at the top. Metadata features and phenotypes are shown as sortable columns across the rows of this multiple sequence alignment, and lastly, hierarchical relations between the sequences are shown in a dendrogram or tree. These encodings are fairly established for genomics visualizations. However, in the current design, we add custom interactions to these integrated views, facilitating multi-reference and multifaceted exploration and the comparison of patterns. Our main design contributions are linked interactions with tailored visual encodings, where sorting and grouping and aggregation showcase the most novelties. We highlight some features here. Users can make selections for highlighting and as input to create groups. For example, it's possible to select one or multiple sequences as a visual reference to compare variations against. The order of how rows appears can be manipulated. They can be sorted by phenotype values, but also by one or multiple genomic positions, which computes a hierarchical clustering of a custom selection, or according to a phylogenetic tree. Being able to sort is essential to identify patterns in these visual settings. All sorting operations are also animated, and the sorting based on categorical values, for example, the one that is based on the nucleotide category of a position, we make minimal number of swaps compared to the previous order, so that the user can keep a mental map. Finally, groups can be created from these selections. They are shown in the same space as individual items to allow for direct comparison at different levels of detail. They can be shown expanded, with or without the highlighting color, or collapsed. The collapsed group shows summary information for the individuals. For displaying the summary information for sequences, we explored several design options. The first attempts mostly focused on displaying distribution information of nucleotides at a position, but because of inconsistencies in shape, size, and position, these hindered pattern search tasks. To help with pattern search tasks, we designed a custom glyph representation where each nucleotide type is shown by a triangle with a fixed position along a clockwise radial axis, and it is colored if it's present in the group. This representation is beneficial for spotting what is similar and what is different, but the main disadvantage is that the encoding hides distribution of the nucleotide types. This distribution is then shown on demand with a tooltip. With our glyph, positions of variation pop out and the patterns with similar content become salient. Genomics experts evaluated our design with 2 use cases. One use case is in Pactobacterium, exploring genotype-phenotype relations in housekeeping genes. The other is in Arabidopsis, where we interpreted SNP markers that may be associated to the important phenotype of flowering time. We now show a simple example of the Pactobacterium use case, where we are interested in finding one or multiple genetic variants that can distinguish one species from the others in this bacterial pangenome. After filtering the unimportant positions, sorting and aggregating the data according to the species category, and then setting the visual reference to the group of interest, a quick sequential browse shows us that there are variants, for example the one on position 399, that can distinguish this group easily from the others. For a thorough walkthrough to the use cases, we refer to our paper and the online interactive demos. So to recap, we presented PanVA, a novel visual analysis design for pangenomic variant analysis. Our main contribution is the customized interactivity combined with a novel glyph encoding to effectively explore patterns in complex pangenomes. Next to the improvements uncovered by use cases and qualitative evaluation, we identified future work opportunities in visual scalability, for example allowing more sequences and larger regions beyond genes, but also in usability, for example naming groups and searching functionality for genes. We have already addressed some of these usability points in an improved code base, which allows practitioners to try the tool with their own data. This is the end of our presentation. We thank all our collaborators who made this research possible and thank you for your attention. For questions, please reach out to me via email.